Hi, my name is Lex Kravitz. I'm an associate professor at Washington University in St. Louis. The topic I'm going to talk about today is using open source methods for behavioral neuroscience in mice. I'm going to show you some devices that my lab has built for quantifying rodent behavior, and I'm going to hopefully leave you with an open source vision of a future for behavioral neuroscience that addresses some of the limitations that have plagued our field for years. I have no conflicts of interest to declare, although I'm going to show you some devices and even point you to some commercial links where you can buy components for them. Neither me nor my lab receives any money from these, and everything is freely available and open source. If you want to screenshot anything during the talk, feel free to do so. The slides themselves are also available at this link. And lastly, I'd like to thank the organizers of San2020 for arranging this conference during a difficult year, and for allowing me this opportunity to present our work. So let's start with the obvious question. Why do we need new behavioral methods? People have been studying rodent behavior for decades. Haven't we figured out the best ways to study it? This is a slide from a review from Brooks and Dunnett showing the diversity of behavioral tests for studying neurodegeneration in mice. Similar slides have been created for many other fields in neuroscience. But despite the number of tests that have been developed, there are still many things we can improve. So what needs improvement? If you talk with people who perform a lot of rodent behavioral testing, and I consider myself one of these people, you will likely hear two common complaints. The first is that behavioral testing is tedious. It takes a long time. As one example, I'm going to tell you about work my lab did with a test called the elevated zero maze. This is a classic test of avoidance of bright open environments. The maze looks like a circle or a zero, and it has two open arms and two protected arms. We place a mouse on the maze and we track where it goes. Normally, mice spend the majority of their time in the protected arms, but if we perturb them in various ways, this balance can change. The mice can become more exploratory, spending more time in the open, or they can become more avoidant and spend even more time in the protected arms. My lab used this test a couple years ago to study the effect of removing dopamine D2 receptors from different populations of striatal neurons. We did this using a floxed D2 receptor mouse that was developed by the lab of Marcelo Rubinstein at the University of Buenos Aires. And what we did was we crossed these mice into different Cre lines to target indirect pathway striatal neurons, cholinergic neurons, or dopamine neurons for removal of the D2 receptor. What we found was that removing the D2 receptor from the indirect pathway neurons, but not the other lines, caused them to avoid the open arms even more than normal mice. So we published a paper detailing why we think this was happening, including many other changes in this circuitry. But that's not why I'm bringing up this data today. What I want to talk about is how long it took to collect the data you're looking at right now. This was 64 mice that were genetic crosses that took months to breed. The test itself was only 30 minutes, but we could only run one mouse at a time with our equipment. So altogether, when I look at this graph, I think about how it represents a huge amount of work, but in the end also represents not very much data. The second criticism you may hear is that behavioral tests are finicky. There's a lot of variance among animals, and many of the relevant factors are difficult to control. Does it matter if we test males or females? If we test them in the morning or the afternoon? House them singly or in group settings? And the answer is that all of this matters, but in practical terms, it is often not possible for us to control these factors, or even understand how they matter to our experiments. Sarah Reardon published an article on reproducibility in behavioral neuroscience called A Mouse's House May Ruin Studies. And it starts with the unhappy lines. It is no secret that therapies that look promising in mice rarely work in people. But too often, experimental treatments that succeed in one mouse population do not even work in other mice, suggesting that many rodent studies are flawed from the start. She goes on to detail the many things about rodent housing that are rarely controlled and often not even reported in our studies, including light cycles, dietary composition, the number of litter mates, all of which can have larger effects on behavior than the study itself. So these are serious problems. But if you think more deeply about the two criticisms I brought up, you may find yourself thinking that these are both symptoms of a different problem. Our behavioral studies are underpowered. I hinted at this in my zero maze example. If I can only test one mouse at a time, does it matter that some were tested in the morning and some in the afternoon? How might this have impacted our results? A lot has been written about this topic. In a widely cited article that I'm showing here, Catherine Button and colleagues analyzed dozens of neuroscience studies and showed that most of the published work in neuroscience is vastly underpowered, which has devastating consequences for reproducibility. This hurts us in two ways. It reduces the chance of detecting a true result when it is there, and it inflates the effect sizes of reported effects, so when people try to replicate the work, they often fail to find the same effect. to look at one figure from this paper. Here they analyzed the statistical power in their sample of neuroscience studies and found that only 15% achieved a statistical power of 80%, which is considered a threshold for an adequately powered study. To throw out some real-world conclusions they make, for a typical water maze study, the authors calculate that the sample size should be about 134 animals, or 70 per group. However, the median number in their sample was 22 animals, about 7-fold lower. So why don't we routinely test 70 animals per group in our behavioral studies? In my opinion, the answer is that most of our labs this just isn't feasible. We don't have the equipment or the time to do this, and even if somebody gave us the equipment and the money to hire lots of people, we wouldn't have the space to house all this equipment. So in this talk, I'm going to tell you about a vision for a new way of running behavioral neuroscience experiments that will allow individual researchers to run hundreds of mice. I'll mention that the main caveat is that this is still a vision. We're not there yet, but I'll show you the progress that we've made towards achieving it. I started by asking, how have other fields solved this power problem? When I look to other fields, particularly microbiology, I find myself envious of their power. They can run circles around their experiments, including replicates, multiple drugs, full dose response curves, all on a single plate. These are designs that would take our rodent neuroscientists months or years to complete. When I see this and think about what we're doing with the zero maze, it feels a bit like we are here. And after months of work, we still haven't even filled up one plate. I also started learning about the progress in addressing this same power issue in other animals, like the ethoscope that Quentin Geisman told us about for studying flies. They can run dozens of fly experiments at one time with a cheap camera. So I wanted to make this, but for mice, and for several reasons that include the space concerns I brought up before, I decided that the best place to do this would be in the rodent home cages themselves. The cages are already there, the mice are already there, so could we just put specialized sensors in their cages and test an entire rack of mice at once? Could the colony rack become the 96-well plate for behavior? So this is what I'm calling a home cage vision for neuroscience. Our lab studies feeding and obesity, so we wanted devices that could measure food intake and also train mice on operant behaviors for obtaining food. Because activity is an important factor in obesity, we also wanted to measure activity levels. And because we learned that temperature and light cycles can impact weight gain, we wanted some environmental measurements, temperature, light, and humidity. And lastly, I wanted it to be flexible and open source, so that when we or others develop new tests, we can easily add them in. To bring up the zero maze one last time, this requires thinking about what are we really interested in with this test? Is it measuring avoidance of light? Or is it measuring the attraction to a safe environment? If we can articulate this more clearly, we can create a sensor for this property and measure it continuously in an entire colony of mice. And then finally, because of the amount of data this will generate, everything should be wirelessly connected to a database for real-time display and analysis. So that's the vision. Now I'll show you how far we've gotten. The first device I'll talk about is the pellet training device, which we call the Feeding Experimentation Device version 3, or FED3 for short. This is an evolution of an earlier device that we called FED that was published by Katrina Nguyen in my lab in 2016. FED3 is a wireless device that contains a lot of hardware. I'm showing a summary here. It lasts about a week on a rechargeable battery. It has a built-in pellet dispenser. It has 2 nose pokes for training mice, tones, lights, etc. It was designed to be placed in the home cage for automatically training mice. Put it in the cage and walk away. We made FED3 open source, and all design files are freely available at this GitHub link. I'm aware of about 30 labs so far that have built FED3. To build it yourself, it costs about $150 for the electronics, and the rest of it is all 3D printed. The GitHub link has instructions on how to build it, as well as videos that talk you through it. It is also being sold by the OpenEFIS production site, which is an incredible initiative for promoting open source resources in neuroscience. We may talk about it more in the discussion and answer period. This is a video of FED3 operating in the cage. So the device is placed in the cage, and the mouse can go poke, and this causes FED3 to play the lights, the tones, and dispense a pellet. FED3 can be used in this way to automatically train mice. This is a graph showing 7 groups of mice representing about 150 animals, all learning to use FED3 for a simple nose poking task. And this graph starts at time 0, meaning that these mice are completely naive to FED3 at this point. We're looking at their first night of experience with it. And over 16 hours, the number of active pokes goes steadily up, In addition, their poke efficiency, the number of correct pokes that they're poking on the side that gives them the pellet, also grows, evidence that they are learning how to obtain pellets from Fed3. While these 150 mice were not run simultaneously like a well plate, like the True Vision, you can still see that having a cheap home cage training device enables this kind of sample size. Because it lives in the home cage, Fed3 can also be used for longer-term experiments. Typically, in operant experiments, animals are trained for 1 to 2 hours per day. In my experience, this is often a function of the equipment availability. Many labs just don't have enough equipment to let one lab member take over all the operant boxes, because other people need to use them. But here, we can give each mouse its own Fed3 device and leave them in the cages for weeks at a time. When we do this, we find that animals learn much more quickly, and we can also observe some interesting things. Here we're looking at 6 days of operant behavior in a group of 10 mice. These mice earn pellets for poking on the active nose poke and earn nothing for poking on the inactive nose poke. We can see that there's a circadian rhythm to their behavior, which is not a surprise, but certainly makes one aware of the perils of testing mice at different times of day. But the surprising thing was that if we look at the percentage of active pokes, the mice were performing a bit more efficiently during the day, meaning that more of their pokes resulted in pellets during their inactive period than during their active period. We also designed Fed3 to be flexible. It has a programmable output that can control external stimuli like LEDs for optogenetic stimulation, or synchronize with brain recordings like fiber photometry. This is a simple experiment replicating a result that we published in 2012, but using Fed to drive optogenetic stimulation of a population of striatal neurons known as the direct pathway neurons. Stimulating these neurons is highly reinforcing, and we can see that mice learned very strongly to respond on the active poke, which resulted in their stimulation. I could tell you a lot more about Fed3, but in the interest of time, I'm going to move on. If you're curious, you can learn more about it at the GitHub site. The next device I'm going to tell you about is an activity sensor. We built this activity sensor and published it last year in eNeuro, which has a new category of papers for open source methods like this. Again, this is open source. All the design files are freely available at the link at the bottom. This work was led by a postdoc in my lab, Bridget Matakainen-Ankney. And Bridget and I decided that as a sensing mechanism, we wanted to use passive infrared sensors. These are the sensors that are typically found in security systems. What's nice about them is that they're extremely low power, so they can run for weeks on a battery. And they have some additional features that make them particularly well suited to sensing activity within mouse cages. So here we're looking at 3 days of data from 8 cages, showing us a clear circadian rhythm. So we can collect activity and circadian data very easily and very cheaply with a device like this. We did validations against video tracking and found that PIR sensing does poorly with small bin widths, but performs well at bin widths greater than 30 seconds. What this means is that if you want to know the distance a mouse is moving in a second-by-second way, PIR sensing is a poor method for returning that. But if you want to know how far a mouse moves every 5 minutes, it's a great choice. It's much simpler than a camera, and in the end, there's only a small data file to analyze. PIR sensing can also be used with any warm-blooded animal. Here's a device tracking a parakeet in our house. You can see that the parakeet is active like us during the daytime. A group in Paris has also been using these devices to track circadian rhythms in lemur monkeys, and they sent us this adorable photo and some really interesting data that they've collected using PIR sensing across weeks with these monkeys. So those are 2 devices that can be used independently. Each one logs data on itself and is completely autonomous. But in my vision, I said we're going to bring everything together and transmit it to the cloud. So in the final few minutes, I want to show you the progress we've made on that. The first thing we did was we started working with a company called MCCI. They're based in New York in the United States, and we worked with them to redesign the activity monitor to be smaller, to contain environmental sensors right on it, and also to include a radio for data transmission to send the data to the cloud. We're calling this device MouseRat, and you can see a demo of the data at mouserat.org. The network side of things is all open source, but it gets complicated pretty quickly, and I don't have the time to discuss each aspect during this talk. But the important things are that this MouseRat device is very small. It fits in a cage. It can accept inputs from devices like Fed, and all the data streams online. So here I'm going to take you on a quick tour through our dashboard. This is a dashboard site known as Grafana, which is free and open source. We've set it up to monitor mouse rats, about 50 of them at the moment. Here I'm looking at 6 months of data. We're going to zoom in on last July. And what we're monitoring is activity levels and pellets coming from fed. Here we're zooming in on one mouse. We can easily export data. It exports to Excel right from the dashboard, but there's Python APIs for grabbing the data automatically into Python. And in addition to the behavioral data, we're getting detailed information on the environment within every cage. So we can see this cage had some increase in humidity. That lasted about a day, and it might be important to know that. We can also look at the light levels within the cage, see when people are opening the doors to the animal facility, that sort of thing. The device is open source. All the design files are available. But as these devices are getting more complicated to build by yourself, it's helpful to have them also sold by commercial partners. So in this case, MCCI is selling it on their store, where you can learn a lot more about the technical specs. You can also check out their GitHub to learn about their Arduino Catena platform, which allows these devices to connect to the cloud. So I'm going to end here. What I want you to remember from this talk is this home cage vision for behavioral neuroscience, which I think is one solution for addressing the problems of throughput and reproducibility in neuroscience. In addition, this cloud-based vision opens up new opportunities for data sharing, for data transparency, for online analyses with real-time results, and I think these things are going to become more and more important in the future. Finally, I'd like to acknowledge the people who contributed to this work. There's a team of people that have been working on FED3 that we continue to develop. There was the team that worked on RAD, which has now kind of evolved into the mouse rat device that we're working on in collaboration with MCCI. And I'd like to acknowledge our funders who made all this work possible. Thank you, and I look forward to talking to you during the discussion period.